Hello students, how are you all? Hope that you all are fine as well as healthy. We are learning 12th Science Biology English Medium, chapter number 5, the principles of inheritance and variation. So in last video lecture of this chapter, we have talked about the chromosomal theory of inheritance. Okay, and today we are going to learn about the different type of aspects which are related or connected with the father of experimental genetics, which is known as the T.H. Morgan, means Thomas and Morgan. So before starting today's video lecture, we are going to revise the last video lectures at a glance. You know that the Mendel, which is known as the father of genetics. Okay, so the father of genetics is known as Mendel. Mendel published a paper, okay, in with different type of data was there which are connected with the genetics, okay, and Mendel did different type of experiments on the pea plant, which is known as the Pisum sativa, and this all data or these records were published in the year of 1865 in the Grun's Natural History Society. And you know that the Mendel was unsuccessful, or the work done by Mendel was not accepted because of the certain reasons, and in these reasons we already talked about this reason in last video lectures. Okay, मतलब पांच छह ना reasons से पांच छह कारणों છે કે શા માટે એનું કામ છે, એનો data છે, it was unaccepted. Then we can say that Mendel died in the year of 1884 after 16 years of Mendel death, means in the year of 1900 scientists republished Mendel work. What are the names of these scientists? First one is Van Schermer, second one is Hugo de Vries, and third one is what? Third one is Carl Correns. Okay, and in the year of 1900, આપણે કહી શકીએ કે એના પાયા genetics ની સ્થાપના establish ત્યાંથી થયેલી. Okay, then you know that the William Batson gave the word which is known as genetics, and Johansen said gene. Okay, Johansen gave the word gene. Effector હતા, બરોબર? Factor Mendel જેને factor કીધા, Johansen શું કહી દીધા એને? Johansen એને gene કહી દીધા. Then in the year of 1900, okay, you know that the cell division was observed with the help of microscope. You know that the one type of development in the process of microscopy. Okay, you know the cell division is for the understanding of cell division was possible, means mitosis, how to understand mitosis and meiosis. Okay, how this all processes, okay, will going to occur. We are able to observe, we are able to understand this all processes under the microscope. Okay, this all were possible during this particular time. Then we can say that two scientists, Walter Sutton and Theodore Boveri, in the year of 1902 said that or observed that the behavior of chromosomes are as parallel as gene. Okay, and after some observation they both gave one conclusion which is known as the chromosomal theory of inheritance. Okay, that's all. And from today we are going to learn about the T.H. Morgan. There is a entry of T.H. Morgan. From today we are going to learn about the work done by T.H. Morgan, means Thomas and Morgan. Okay, so see, I am writing the one name which is known as T.H. Morgan, means Thomas and Morgan. T.H. Morgan means you can say that Thomas and Morgan. T.H. Morgan means you can say that Thomas and Morgan. So first point we can say that Thomas and Morgan is known as father of experimental genetics. You can say that Thomas and Morgan is known as father of experimental genetics. So you remember, Thomas and Morgan is known as father of experimental genetics. We can say that he and his co-workers did different type of work. Okay, we can say that they gave they means he means Thomas and Morgan and his co-workers. They gave experimental proof. They gave experimental proof of chromosomal theory of inheritance. Chromosomal theory of inheritance. So you remember that the experimental proof, okay, of this theory. You know that this theory was given by Walter Sutton and Theodore Boveri, but the experimental proof of this particular theory was given by T.H. Morgan and his co-workers, and they also gave one proof that the reason for variation is nothing but it is sexual type of reproduction. It is nothing but it is sexual type of reproduction. Okay. It is what? It is sexual type of reproduction. So you can say that the reason for what? The reason for variation is nothing but it is what? It is sexual type of reproduction. Okay. So it is about the T.H. Morgan. 2 points are there. First point is about he is known as the father of experimental genetics, and second point is about the what? Okay. First one gave experimental proof of the chromosomal theory for inheritance, and second one we can say that the reason for variation is what? The reason for variation is hidden in the sexual type of reproduction. Then point number 3, we can say that T.H. Morgan, he did different type of experiments. He did different type of experiments on a particular organism which is known as fruit fly. See, we are not talking about house fly. Okay, house fly means Musca domestica. We are not talking about the house fly. We are talking about the fruit fly, and the scientific name of fruit fly will be Drosophila melanogaster. See, underline is must because it is one type of scientific name. Genus and species, both names are included in this particular name. So underline is must. Okay. So third point is very much important. T.H. Morgan, Thomas Hunt Morgan did different type of experiments on a particular fixed organism which is known as fruit fly, Drosophila melanogaster. Mendel ma pea plant, Pisum sativum hatu, and Morgan ma fruit fly, Drosophila melanogaster. Now we are talking about some reasons. We are going to talk about some reasons that why Morgan selected Drosophila or the fruit fly for his experimental work. So we can say that first reason is what? We can say that it is easy to cultivate or culture. It is easy to cultivate of fruit fly in a synthesized medium in a laboratory. So first point is what? We can say that it is easy. It is what? It is very easy to cultivate, okay, of fruit fly. Means cultivation of fruit fly is very much easier. તમે એને લેબમાં ઉછેર કરી શકો. કેવાનો મીનિંગ એ છે? The cultivation of fruit fly is very much easier, okay, in a synthesized means prepared medium in a laboratory. Okay, this is the first reason. Second reason we can say that the wall's lifespan will be of the 2 week. Okay, the wall lifespan of wheat will be 2 week. We can say that after one mating means you can say that the process of reproduction, population, we can say that after one mating large numbers of progeny large numbers of progeny will be formed, okay, or will be produced. मतलब जो તમને generation માં result ઝાઝા બધા મળે છે, means you are able to compare the result of the generation or the character of generation with their parent. Means you can say that after one mating, after one population, you are able to get the maximum numbers of progeny. It is the third reason. What about the reason number 4? Reason number 4, we can say that sexes are separated. Sexes are separated. Means these are dioecious type of animals, and sexes are separated. Because of it, we are easy to identify particular male organism as well as female organism. Means we are able to separate, we are able to identify the male fly and female fly. Okay, point number 6, we can say that under low microscope we are able to separate the different type of characters. Okay, it's easy to observe under the low microscope or with the help of naked eyes. So these are the 6 reasons. Okay, so the first part of today's lecture is completed. Again, we are going to revise this. See, Thomas Hunt Morgan, which is known as the father of experimental genetics, he and his colleagues, okay, his colleagues did work, different type of work. The first one is they gave the experimental proof of the one theory which is known as chromosomal theory of the inheritance, and they said that the reason for variation is there in the sexual type of reproduction. Then he did different type of experiments on a one particular organism which is known as fruit fly, and the scientific name of fruit fly is Drosophila melanogaster. What about the different type of reason? Means why Morgan had selected this particular organism? Because we can say that it is easy to cultivate of fruit fly in a synthesized medium in a laboratory. Point number 2, we can say that lifespan will be of 2 weeks. Third one, after one mating, large number of progeny will be produced. Sexes are separated. That's why it's easy to identify the male fly and female fly, and we can separate or we can observe the different type of characters, means different type of variation with the help of naked eyes or with the help of microscope, okay, or with the help of low powered microscope. These are the reasons. Okay. Now we are going to talk about the part number 2 of today's lecture. Very important lecture, most important question, and the title of question is linkage and recombination. Linkage and what? Linkage and recombination. Okay. What is the title of question? Title of question is the linkage and recombination. It is a question of 3 marks. Okay, for 3 marks. For 3 marks, it is the most important. It is the most scientific question of 3 marks. Linkage and recombination. See here. खाली તમારે બે વર્ડ નો મીનિંગ જ સમજવાનો છે. Okay, see here. First point, we can say that T.H. Morgan and his co-workers, his colleague, did different type of experiments on fruit fly, means Drosophila melanogaster. Any word also fruit fly workers. Okay, now point number 2. If we talk about the dihybrid cross, okay, dihybrid cross, then Morgan did same dihybrid cross which was done by Mendel. And what about the result of dihybrid cross? You know the result is 9:3:3:1. Okay, 9:3:3:1 is the result of the dihybrid cross. Okay, of F2 is the result of F2 generation of dihybrid cross which is done by Mendel. Okay, yes or no? So the same experiments were done by T.H. Morgan. Okay. Point number 3, we can say that Morgan take one female fly and one male fly. SP generation. So as parent, SP generation, Morgan selected one female fly and one male fly. You know that sexes are separated. This female fly having yellow colored body, or the body of this fly is yellow colored. Okay, and if we talk about the color of eye, then this fly having white colored eye. So બે character છે કે નહીં? Dihybrid cross means inheritance of two genes. Okay, so for inheritance of two genes, two characters are selected. Here we can say that if we talk about the color of body and if we talk about the color of eye, okay, then we can say that here red colored eye is there, and here brown colored body is there. Okay, two characters are there. Yes or no? And if we talk about the, હવે આમાં એક્ઝામ્પલ કઈ ગણવા નથી. If we talk about the F1 generation, then મેન્ડલે શું કર્યું F1 માં? તો કે સેલ્ફ ફર્ટિલાઇઝેશન કર્યું હતું કે નહીં? Yes or no? તો સેલ્ફ ફર્ટિલાઇઝેશન was done in F1 generation. મોર્ગને પણ શું કર્યું? એ જ કર્યું, સેલ્ફ ફર્ટિલાઇઝેશન. But what about the F2 generation? We can say that in F2 generation accepted result is what? સમજો, in F2 generation result તમને કંઈક આવું મળવું જોઈએ કે નહીં? result તમને 9:3:3:1 મળવું જોઈએ, મેન્ડલ જો સાચા હોય તો. but result was not similar. okay, or result is not like this. result આવું ન હોય, મતલબ કે મેન્ડલ આ કેવા પડ્યા? ખોટા પડ્યા. મોર્ગને શું કર્યું? મેન્ડલ જો બે કેરેક્ટર બોડી યલો, બ્રાઉન, આઈ વ્હાઇટ, રેડ, એક ડોમિનન્ટ હોવું જોઈએ, રિસેસિવ હોવું જોઈએ, તો રિઝલ્ટ આવું મળવું જોઈએ ને. આવું રિઝલ્ટ નથી મળ્યું. કોને? TH વર્ગનને. ગોટી પેલો પાર્ટ પૂરો. હવે તમે પાછો અહીંયા સ્કીપ કરો. બીજો તમને એક મહત્વનો કોન્સેપ્ટ સમજાવી દઉં. એક હું લખું છું પેરેન્ટલ જીન. first one is parental gene, and second one is the non-parental gene. चलो आ બે કોન્સેપ્ટ સમજીએ, પેરેન્ટલ જીન એન્ડ નોન પેરેન્ટલ જીન. તમે એમ કહો છો કે if we talk about the P generation of dihybrid cross, capital R, capital R, capital Y, capital Y cross with small r, small y, small y, it's phenotype. What about phenotype? Phenotype is round and yellow, and here we can say that wrinkled and green. Okay, wrinkled and what? Wrinkled and green. If we talk about the result of F2 generation, yes, result is what? Result is 9:3:3:1. તમને રિઝલ્ટ આવું મળ્યું, 9:3:3:1. 9 તમને કેવા મળ્યા? તો કે રાઉન્ડ એન્ડ યલો મળ્યા. 3 કેવા મળ્યા? તો કે રાઉન્ડ એન્ડ ગ્રીન મળ્યા. 3 wrinkled and yellow, અને બાકીનું પણ wrinkled and green. Dominant is what? Of course, dominant is the round and yellow. હવે જો રિઝલ્ટમાં તમને 16 માંથી 9 વખત કેવું મળે છે? રાઉન્ડ એન્ડ યલો મળે છે. 16 માંથી 9 વખત રાઉન્ડ એન્ડ યલો. આ કેવા થઈ ગયા? ઇટ્સ ધ પેરેન્ટલ જીન. ઓકે, આ તો રિસેસિવ છે, આ જમાના જ નથી. રાઉન્ડ એન્ડ ગ્રીન પેરેન્ટ્સમાં છે જ નહીં. ઇટ ઇઝ નોન પેરેન્ટલ. ઇટ ઇઝ ઓલસો નોન પેરેન્ટલ. આ રિસેસિવ છે એટલે લખતો નથી એને કંઈ. ખબર પડી? આ પેરેન્ટલ છે જીન, આ ધીસ બોથ આર નોન પેરેન્ટલ છે. પેરેન્ટલ જીન વધુ વખત મળે છે, મતલબ વધુ વખત ભેગા રહે છે. 16 માંથી 9 વખત ભેગા રહે. વધુ વખત ભેગા રહે મતલબ કે ઓછી વખત સેગ્રીગેટ થાય છે. ગેટ ઇટ? ઓછી વખત શું થાય છે? જુદા પડે છે. ખબર પડી કે નહીં? ગોટ ઇટ? યસ ઓર નો? સો વી કેન સે ધેટ there is a one type of linkage or bond between parental gene. Got it? Yes or no? There is one type of linkage or bond between parental gene. It means now we are talking about the linkage. But before starting this, I am writing the one point. TH Morgan and his colleagues said that all the genes are located on X chromosomes only. जो आ છે ને એ वन लाइनर क्वेश्चन છે. આ લોકો એમ કહે છે ખાલી બધું જીન ક્યાં આવેલા છે? ઓન્લી ઓન ધ X ક્રોમોસોમ. Y ક્રોમોસોમમાં કઈ છે? જો પેજ કરો ડાયાગ્રામમાં Y ક્રોમોસોમ બતાવ્યો છે તમને ગ્રીન કલરમાં. એ Y ક્રોમોસોમમાં જીન બતાવ્યા જ નથી કેમ કે આ લોકોના મત મુજબ all genes are located only in X chromosome. Okay, now point number 2, we can say that there is a one type of physical connection, or there is one type of physical bridge between the different type of parental gene. There is a one type of physical bridge or physical combination, physical bridge, okay, between the parental gene. It is known as what? It is known as linkage. Linkage તમને કેમાં જોવા મળે? Linkage તમને parental gene માં જોવા મળે. હવે સમજો, તમને ખબર છે ને સિનોનિયમ્સ એન્ટોનિયમ્સ આવતા, દિવસ રાત, ડે તો નાઇટ, સુખ તો દુઃખ, બરોબર છે, ભાઈ તો બેન, બ્રધર સિસ્ટર, એમ. Linkage to recombination. We can say that recombination is always found in the non-parental gene. Parental gene માં જો linkage જોવા મળે તો non-parental માં શું જોવા મળે? Recombination જોવા મળશે. Take it clear? Yes or no? Now another example. આ પાછો મોર્ગનનું વાત ચાલુ છે. મોર્ગને પાછું એક એક્ઝામ્પલ આપ્યું. એને એવું લીધું, one fly which having the white color and yellow color gene, and second example, one fly which having the white color and miniature, એટલે પાંચ નાની હોય તો miniature. પણ એ પેલા તમને કહી દઉં, suppose it is a one chromosome, then if સમજો, if capital R and capital Y, જોવા જઈએ આ બે એકબીજાની નજીક છે કે નહીં? હા પાડો, નજીક છે કે નહીં? તો યસ, નજીક છે. નજીક હોય લિંકેજ વધુ, રીકોમ્બિનેશન ઓછું. આ જોવા જઈએ સ્મોલ r સ્મોલ y, એકબીજાથી દૂર છે કે નહીં? હા પાડો, દૂર હોય લિંકેજ ઓછું, રીકોમ્બિનેશન વધારે. યાદ રાખજો. અહીંયા if we talk about directly F2 generation, we can say that the recombination will be 1.2 or 1.3. Then linkage will be 100 માંથી માઈનસ કરી નાખો, 1.3 રાખે 98.7%. જોવા જઈએ આ બેનું ડિસ્ટન્સ કેવું હશે તો બતાવ્યું હશે બુકમાં. આ રીતે આ સેન્ટ્રોમિયર છે. અહીંયા કીધું છે ડબલ કીધું છે વાઇટ. જોવા જઈએ ડિસ્ટન્સ ઉપર છે, ડિસ્ટન્સ ઓછું, recombination ઓછું, linkage કેવું છે વધારે છે. એટલે આ બેનું સેગ્રીગેશન ઓછી વાત છે. અહીંયા recombination will be 37.2%. linkage 100 માંથી માઈનસ કરો એટલે 0.8, 38, 62.8% linkage જોવા મળશે. બરોબર છે. અહીંયા ડિસ્ટન્સ વધુ, recombination વધુ, linkage ઘટી. એટલે આ યાદ રાખજો. if linkage is maximum then distance will be minimum. If distance will be minimum then recombination will be maximum. If distance is maximum then linkage will be minimum. If linkage will be minimum, recombination will be maximum. આ ચાર આંકડા તમારે યાદ રાખવાના છે. લાસ્ટ પેરેગ્રાફ છે, તમને ખાલી બોલીને કહી દઉં, લખીશ નહીં. બરોબર છે? પેલા તમે જો સ્ક્રીનશોટ લઈ લો આનો, બોલાઈ ગયું છે કઈ ગયું. Take a screenshot of this, okay? Last paragraph is about the Alfred Sturtevant. There is a one named Alfred Sturtevant. એક નામ છે Alfred Sturtevant. એ મોર્ગનના શિષ્ય છે, student of the CH Morgan. Okay, he used the frequency of gene. બરોબર છે? frequency of gene in the genetic mapping. મેપિંગ માટે એને linkage અને recombination નો યુઝ કરેલો છે for the mapping, and this mapping is used in the human genome sequential project, HGP. You can say that HGP means what? Means human genome sequential project. HGP means human genome sequential project may be used. So three important words: Alfred Sturtevant, linkage, recombination, frequency of gene. In HGP means human genome sequential project. These three words are very much important. Got it? Yes or no? So today's lecture is completed. Again, we are going to revise today's lecture at a glance. We can say that the T.H. Morgan and his colleagues did or gave the experimental proof of the chromosomal theory of inheritance and said that the reason of variation is nothing but it is sexual type of reproduction. Then we can say that what? Fruit fly, Drosophila, melanogaster, and reasons are there. Linkage and recombination. જો આ પહેલા એવું થયું સમજો મોર્ગન છે ને એ મેન્ડલે જે કામ કર્યું એમાં જ હાલતા હતા એ જ વસ્તુ કરતા હતા પણ અહીંયા એને રિઝલ્ટ ન મળ્યું ને એટલે પાછું કાંઈક એને નવી શોધ કરી ખબર પડી હું શું બોલું છું એટલે લિંકેજ અને રિકોમ્બિનેશન ની શોધ થઈ એ લોકો એવું જ માનતા હતા કે એક્સ ક્રોમોઝોમ ત્યારે જો સમજો મેન્ડલ વખતે ક્રોમોઝોમ હતા જ નહીં ક્રોમોઝોમ તો મોર્ગન વખતે આવ્યા બરોબર છે એટલે એ શું કહે છે વાય નું કઈ કામ નથી ખાલી એક્સ ક્રોમોઝોમ જો તમારી બુકમાં વાય ક્રોમોઝોમ ગ્રીન કલરમાં ખાલી આપ્યો છે આવું લખેલું હશે તમે જોજો આ માહિતી છે ને વળેલું ક્રોમોઝોમ બતાવ્યો છે ઇટ ઇઝ અ વાય ક્રોમોઝોમ એનું પર જીન જ નહીં બતાવ્યું એટલે યાદ રાખજો પેરેન્ટલ નોન પેરેન્ટલ બરોબર યાદ રાખજો ઓકે લિંકેજ વધુ મતલબ કે સેગ્રીગેશન ઓછું થાય આવો ક્વેશ્ચન કહે છે આટલું સમજાય છે પછી તમે બુકમાંથી વાંચજો હવે બુકમાંથી આવડી જશે સીધું ટેક્સ્ટ બુકમાંથી વાંચવાનો પ્રયત્ન ન કરતા તો નહીં આવડે લેંગ્વેજ થોડી ઘણી હાર્ડ છે ઓકે સો ટુડેઝ લેક્ચર ઇઝ 100% completed. I hope that you are getting it very much easily. Thank you very much.